The final approach, total protein staining, is becoming the new gold standard for normalization. Concerns about the stability of housekeeping protein expression, stripping and reprobing issues, and antibody availability for signaling proteins have prompted a search for alternatives that can streamline the process and reduce variability. At the end of 2015, the Journal of Biological Chemistry issued recommendations for normalization, stating that normalization of signal intensity to total protein loading assessed by staining membranes using Kumasi Blue, Ponceau S, or other protein stains is preferred. If we review the qualities of an accurate internal loading control and compare them with this method, we see that endogenous proteins present in all samples is key in this approach. Total protein staining is a direct measure of the total amount of protein in each lane. There is no indicator internal loading control. An increase in protein concentration will result in a linear increase in the signal intensity as long as the membrane and the bands are not saturated. Like the signaling protein strategy, the change in expression due to experimental conditions is inherent in the samples and is already accounted for. Finally, as with the other forms of normalization, you do have to verify that the total protein is detected within the same linear range as your target protein using your specific target antibody or antibodies, sample concentration, and detection method. However, the total protein staining itself is antibody independent and does not require validation for each experimental context or condition, the biological system, and primary antibody. There are several different ways to apply this method. One way is to stain a replicate gel for comparison with Kumasi Blue, for example. A second way is to apply stain to the membrane prior to immunodetection. An example of this would be revert total protein stain. A third way is to add the stain to the membrane post-immunodetection, such as with Kumasi Blue. A completely different twist is to label the proteins prior to or during running on the gels with a detection moiety, a so-called stain-free method. The way you choose to apply this method comes with its own set of benefits and drawbacks. Let's look at them one at a time. Staining a replicate gel for comparison is an excellent method for standardization of sample loading. It does nothing, however, to address potential inconsistencies that can occur during transfer. Direct staining of the membrane either before or after immunodetection gives you the total protein concentration for each lane and accounts for the associated transfer variation if present.